Okay, could somebody confirm that you can hear me?
I can hear you.
Thank you. All right, are there any questions? There's no questions. Let's begin. And let me wish you a happy Thanksgiving. I don't know. Can you guys see my screen now? I've got another message saying that Microsoft is having trouble verifying my account with this computer. And they're going to shut down my access to use Microsoft starting Saturday, November 26th, which is really bad because that means I'll have to go in tomorrow because that would be the only possibility I could get my computer looked at before Thanksgiving, before they shut it down. I clicked on the get help and it didn't give me much help. So anyways, we're going to be starting chapter 13. You're going to have to live with my voice. I'm not sure why it's a little hoarse today. We did have a change in the weather. I don't feel sick or anything, but I was out in the weather and got up very early to take in my car today. And so that might have something to do with it. Anyways, please bear with my hoarse voice. And we're going to be discussing chapter 13, viruses, viroids, and prions. Let me show you the syllabus. Blow that up a little. There's no class, of course, on Thursday, and there will be I don't know, slightly a lab meeting. I will be there from 6:30 to 6:45 to answer questions. Once the last question is answered or it becomes 6:45, I will log off. There's no lab or lecture on Thursday because of Thanksgiving. Any question about any of that? If not, we'll begin chapter 13, and notice we will finish chapter 13 after Thanksgiving. So my goal is only to get about half of it done. In the lab, there is no lab today, but you should be working on your unknown project. Even if you've identified your unknown, you should be working on your report. Okay. And I will try and get a lot of grading done this Thanksgiving weekend, but I'm not sure I'm going to get a lot done, particularly tomorrow. I've got a lot of baking and cooking to do and planning to do. And then on Thanksgiving, there's the get together. So I'm not sure how much time I'll be able to get in grading on either of those 2 days, but on Friday and Saturday, hopefully I can get more grading done. I did do a little bit of grading, and you can take a look at it. I don't remember what I got graded, but last week I got at least one quiz graded and several homework assignments graded. Oh, and I got some of the labs graded for the second time. I think labs 0 to lab 4 are graded for the second time, if I'm remembering correctly. All right, any questions on what we're doing? If not, let me go ahead and minimize that. And we'll be discussing chapter 13, viruses, viroids, and prions. As I always do, I give you a first slide, which gives you the major goals of the chapter and a rough outline that we will roughly follow. So, of course, always know the terms for the chapter. In this term, we have several: capsid, envelope, spike. All of those refer to a virus. So does actually bacteriophage. And then the lytic and lysogenic life cycles. Know what a prophage is, a provirus. and then two more life cycles, the latent life cycle and the persistent infections life cycle. And then lastly, we'll talk about prions and viroids. Any question about any of that? All right. Understand the characteristics, the structure, and the classification of viruses. Understand about viral multiplication, and they multiply in a life cycle, and we'll discuss that. And then lastly, understand what are prions and viroids. Any questions? If not, let's turn to talking about viruses. The only goal a virus has is to replicate. And how do viruses replicate? They can't replicate on their own. They have to have a host cell to replicate. And what the virus does is it infects the host cell. That's the cell it grows in and replicates in. And the virus invades the cell and then hijacks the cell and forces the cell to make viral copies. And then these viral copies escape from the cell. Any question about any of that? Let me see if I can get this to work. It used to be they didn't work, but I think Microsoft changed this, and now PowerPoint is launching a web page. No commercial. This is YouTube.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any questions? So let's talk about some of the general characteristic of viruses. Viruses are inert when they're not within a host cell. That means they have essentially no metabolic activity. Excuse me, frog in my throat. No metabolic activity. They're not really alive. So, well, that's debatable, but they don't have any activity as if they were alive outside of their host cell. And they do not replicate outside of their host cell and obviously have no metabolic activity outside their host cell. They require a living host cell to multiply. So we can say that viruses are obligate intracellular parasites, a parasite that must have a cell, meaning obligate, in order to reproduce and actually live. if live's the correct term. Viruses multiply within living cells by using the host cell's enzymes, nucleic acid, amino acids, and other machinery within the cell, the other contents of the cell. For example, viruses have no ATP generating mechanism, and yet they need ATP. And how do they use ATP? Well, they use the cell supply of ATP. Viruses do have a genome, and this genome consists of either DNA or RNA. Note the term or, meaning that the genome is neither both DNA and RNA. It's either one or the other. Besides the genome, viruses always have a protein coat which surrounds the nucleic acid. So the nucleic acid, the genome, is the innermost molecule of a virus. And at the very minimum, a virus also has the genome surrounded by a protein coat. Some viruses have other, parts to it, like some viruses have an outer lipid envelope, which is on the outside of the protein coat. We call the protein coat a capsid. Any question about any of that? Some other characteristic of viruses, they have a host range. This is the range of cells that the virus can infect. The host range is determined by specific host attachment sites. So the host cell has to have attachment sites that we call receptors. And then the virus has attachment molecules that are sometimes called ligands. And I won't quiz you on that because the attachment site does have different names depending on, I guess, what the author is of your textbook. And, I'm not sure what your textbook author uses. Sometimes they're called adhesions, meaning sites of the virus that attach to the host cell. But always the host factor that the virus binds to is called the receptor. Most viruses only infect the cells of one species. And even if they do infect more than one species, most viruses only infect like related species. For example, a HIV virus can only infect the cells of two animals, humans and one of the chimpanzees. If you don't know, there are actually two species of chimps, and one of them can be infected by HIV. However, the chimp does not develop AIDS. The virus does not destroy the chimp's immune system. Any questions about any of that? And further, when we look at HIV, besides infecting the cells of only two species, they only infect certain cells, like obviously T cells, T helper cells, a big one. And they do infect, I think, macrophages and brain cells and a few other cells. And that just tells you how specific many viruses are. Now, not all viruses are that specific. For example, if you don't know, the flu virus can hit humans and can hit horses and can hit pigs. Okay. And what do you notice that's common about humans, horses, and pigs?
They're mammals.
Yeah, they're all mammals. And not only that, they're either omnivores or herbivores, meaning the flu doesn't usually hit carnivores like cats and dogs. Most of the time do not come down with human flu. And it's just that viruses are very specific. That isn't always the case. There are some viruses. The flu was an example of it hits more than one species, but it's also specific to mammals. But there are other viruses which hit warm-blooded animals like birds and humans and mammals. And I'm trying to remember the name of this one. I'm losing my mind, so I'm not remembering. What the heck is that name of that virus? It's the one that was hitting the spotted owl. Nobody knows. It just recently came to the United States within the last 10 years, maybe 15 years. Oh well, it'll come to me. Anyways, that one can hit almost any warm-blooded animal, including birds and mammals. And then some viruses like I think it's Rocky Mountain spotted fever can live in ticks as well as humans and many other mammals. In which case, some viruses have a broader host range, much broader than most viruses. But most viruses are very specific, infecting only about the cells of one species. And the reason for the specificity is the receptor on the host cell, and then the adhesion molecules or ligands on the virus have to bind to each other. And so only a narrow range of cells can be infected. For example, viruses that hit bacteria are not going to be infecting your cells. And then, of course, vice versa, viruses that infect your cells are not going to infect bacteria. Bacterial viruses are called bacteriophages, and sometimes that's shortened to phages or phages. And I will tend to use bacteriophages to be very specific about what we mean. Any question about any of that? All right. When we're talking about viruses, they have a very specific size, and that is they're much smaller than a eukaryote cell, and they're smaller than a prokaryotic cell as well. So here we have the size of a red blood cell, which is actually a fairly small eukaryotic cell. And then we have the size of a E. coli, which is a bacteria that's a little longer than your average bacteria. The width is about the correct size of an average bacteria. And the longest virus does approach the size of a prokaryotic cell. In fact, it's bigger than the smallest prokaryotic cell. We won't talk about what the smallest prokaryotic cell is, but viruses, their length is bigger than the smallest prokaryotic cell. However, their diameter, which is actually the megavirus has the biggest diameter, is always smaller than a prokaryotic cell. Is that true? The largest virus approaches the diameter of the smallest prokaryotic cell. And then viruses, of course, can get smaller than that. This is the largest virus. That's the longest virus. The smallest virus you've ever heard of is probably the poliovirus. You can get a virus smaller than the poliovirus, such as bacteriophage MS2. But how many of you have ever heard of bacteriophage MS2? Anyone? I didn't think so. But probably all of you have known the poliovirus, and that's the smallest virus that you're aware of. Oops, sorry, that's the poliovirus right there. So for the largest virus, its diameter is about 440 nanometers. That is 0.44 micrometers. And then virus, the smallest one, reaches about 20 nanometers in diameter. Any question about any of that? All right. When we're talking about viruses, we have a specific term for a virus particle. A new virus particle is called a virion, a single mature, complete infectious virus particle. So I once told you that viruses invade a host cell and then use the host cell to make copies of itself. The new viruses that the original virus makes are called virions. So to be a virion, you have to have everything necessary, at least the genome, necessary for the virus. A single mature virion is a complete infectious virus particle, and it does have a genome. If it didn't have a genome, it would not be, what's to say there, infectious? The way viruses replicate, that's true, although that'll be kind of confusing to you at this point. Any question about that before I go on? All right. So I mentioned that a virus has a genome. The genome is made up of nucleic acid. The genome is either DNA or RNA. It is never both. When we're talking about an RNA virus, it's more important just to talk about it's an RNA virus. You need to talk about whether the RNA virus is double-stranded or whether the RNA virus is single-stranded. And SS, lowercase ss, stands for double-stranded, capital P, lowercase s. Did I say that right? Now I've lost my train of thought here. The genome of a virus can be either double-stranded or single-stranded. That's what I was talking about. RNA viruses. It's important to discuss whether an RNA virus is a double-strand RNA virus or a single-strand RNA virus. And if the virus is a single strand or its genome is a single strand of RNA, then it's important to state whether that single strand RNA is the positive single strand or the negative single strand RNA. As you know, when we're talking about DNA, which in human cells is always double-stranded, not always, it's normally double-stranded, it's single-stranded during replication and transformation. You know about double-stranded DNA. Double-stranded RNA is the same. It's just that it's RNA instead of DNA. But it actually hydrogen bonds between the bases of each double-strand RNA. Now, let me state that in nature of eukaryotic cells, we do not see double-stranded RNA. Only in viruses do we see double-stranded RNA viruses. Any question about any of that? So in summary, the RNA, no, the viral genome can be either RNA or DNA. If it's DNA or RNA, then you have to divide the virus up. And usually you divide them by a clump, by family if you need to, by genera if you need to, and then by the characteristics of the virus. So that a single-strand DNA and a double-strand DNA virus are very similar, essentially the same. The difference is that double-strand DNA has a bend. Let me see if I still have that here. I don't. Has a bend which gives structure to the DNA. And the single-strand DNA will do something similar. Instead of two strands wrapped around each other, it's going to be a single strand wrapped around each other. Any questions about the genome of viruses? We can have double-stranded DNA genome and single-stranded DNA viral genome. Both of these should look familiar to you because we did discuss how the genome of a virus replicates. And at least for the viruses that we tend to be studying, trying to remember the name of the virus that we're going to look at, it's something like CAF. I don't remember what CAF stands for. If the RNA, if the genome of the virus is RNA, then you need to break it down into double-stranded RNA, and that's when the RNA will be bound together in hydrogen bond between the two strands, and then the substrate can come in and open up that double-stranded RNA genome, and it's where there will be a bubble is where the virus will infect the cell. Maybe infect isn't the quite the correct word because the viruses aren't making a, excuse me, the penguin isn't making a parasite. The penguin isn't eating or living off of the parasite, living off of the virus. Did you guys get that? I just made another mistake. I'm a little tired, so I'm probably going to be error prone today. Any questions about what I've talked about so far? All right. So if the viral genome is an RNA genome, and if it's single-stranded RNA, then you need to go further and say whether the single-strand RNA is double-stranded or is the RNA side of the box coated with cheese. You have to state whether the virus, if it's RNA and single-stranded, whether it's negative strand, single-stranded. So you got two types of single-stranded RNA viruses. You have the positive single-strand RNA and the negative single-strand RNA. The two viruses are together, I'll draw it here. I'm running out of space. So if we had RNA, and let me think it's AUG, for example. And I don't know the next, so we're just going to go like that. If it's single-stranded, you need to state whether it's positive single-stranded or negative single-stranded. For this virus here, if the corollary were U, A, C, whoop, I missed the letter. UAC, got my caps lock on, that's the problem. Then this one would be the single-stranded RNA, positive strand, and then the other one, where did I lose that? There it is, would be negative strand, meaning not facing the main street, and well, the single-strand RNA can be one of two strands, positive strand or negative strand. They are similar, meaning that base pairing will occur between the negative strand and the positive strand of RNA. And that's why we get double-strand RNA, because the single-strand RNA comes together and then doubles, acting like DNA. But viruses do not need double-stranded DNA to replicate. They can replicate with other means, such as COVID-19 or Mycobacterium tuberculosis. All right, if there's no questions, let's go on out to your, is it a dig? Or is it planted in fields? You guys probably don't know because you've never gone on a field trip. What I should do is make a virtual field trip, but I don't, so you're going to have to listen to me talking about it. The difference between the single strand positive stranded RNA and the negative single strand RNA is that the RNAs, of course, will run in opposite direction. I don't know if you can see this, but I'm trying to make them run in opposite direction. And the single strand RNA can also give the cell a new phenotype. For example, Aman Roca, which hopefully you guys are familiar with. It's not alive, so this isn't a good example, but we'll pretend it's alive. It can be in a specific environment and then replicate. At least some of the replication genes will be turned on. Why does that happen? Because the beaverton is exposed to, I'm not sure how you say this, Xinghu, something like that. All right, any questions about single-strand RNA and the positive single-strand RNA and the negative single-strand RNA? All right, let me go back to double-stranded RNA just for a second and state the double-stranded RNA is unheard of in our cells. Eukaryotic cells do not normally have double-stranded RNA. However, virally infected cells can have two strands of viral DNA. In fact, many companies will actually do more than one or two, and I don't know what the maximum number is. The positive single-strand RNA can be read by the ribosome. And so the ribosome will move down the single strand, and once it hits the lettuce, the hole for the lettuce will fall in, and then we're done, although I need to take up my groceries and pay for them, which I have done, but I'm saying besides that, we're done. Any questions about DNA or RNA, but not both? The DNA of nucleic acids, as well as the older individuals who are more likely to come down with COVID-19. The COVID-19 can stick to the pan, although if you boil it, you'll kill the COVID-19. And you could provide some protection, not all protection, but at least some protection by turning on the lights. If your college, which as far as I know, Clark College does have a generator, you can hook the generator up to the windmills and then rotate them and have fun watching them. Any question about any of this? All right. Viruses can have their genome in a linear arrangement, or they can be a circular arrangement. Now, as you know, when we were talking about eukaryotic cells, viruses contain their genome only in a linear segment of the chart, whereas the circular virus can be both linear and you can never replace it and slowly watch the building collapse. All right, any question about any of this? I've already talked briefly about the capsid. It's just I didn't maybe mention it as the capsid. The capsid is only on some viruses. And if the capsid is attached to the cell wall, then the virus will affect the CAR. If the CAR has signs or symptoms, then disease can result. When we're talking about viruses, I mentioned earlier that viruses have a protein coat. Another name for that is capsid. Capsid proteins can be the same virus, which I actually showed you just a little while ago. Here? No. So viruses do need DNA or nucleotides to build the virus genome, the new virus genome. And you can scavenge from other CARs to do that. However, viruses have no ATP generating mechanism. And for their ATP needs, they get their ATP from their host cell. All right, any questions about any of this? I think I'm going the wrong way. I should probably get rid of those. We'll talk briefly about that later, but for the moment, I want to define viruses. They always have an internal genome. Viruses, and this is true of all viruses, also have a subunit that we call a capsomere. And capsomeres are protein that can be the same or different proteins in the soil. For example, in the past, I grew mushrooms, and you have to provide a specific environment so the mushrooms will continue to grow. And I got my mushrooms growing for about 6 months. I think what I did was I took the viruses into school and then forgot about them once I brought them home. And so the viruses died out in the salami. And if you don't know, salami is fairly salty, so is pepperoni. And so you don't need to add much. Don't need to add much fruit to the meal. Okay, any questions? All right, the capsid, and remember that's the protein coat surrounding the virus. The capsid can be composed of subunits that we call capsomeres. Viruses that have capsids can also secrete a substance which goes out generally further than the capsid. And this substance is really wet and essentially invisible. I don't know if anyone noticed it, but the virus that we were talking about, we talked about 2 of them, can show up in the, what do you call that? The football. So all viruses have a capsid. Not all viruses have DNA or RNA or both. Let's move on. Some viruses have an envelope surrounding the protein coat that we call the capsid. And it's composed of capsomeres. Oh, let me show you a capsomere. Come on. Yeah, I can't get it to move. Let's see if that works. That worked. Oop, too far. All right, so there's the virus genome, the innermost molecule, and here's the capsid, also called the protein coat of a virus. This part is cut away so we can look inside of it. The envelope would be outside of the capsid. And what else did I want to talk about? Oh, so the capsid can be the same protein as it is here, where the protein coat surrounding the viral genome is essentially only one protein. That's not always the case. Sometimes viruses can use many cell can openers. And especially if you cook at home, you'll use many more natural ingredients for your own health. I'm not going to talk about the health, but generally you need fiber and fruits and vegetables. Otherwise, you'll be unhealthy. All right. If a virus has an envelope, we call it an enveloped or enveloped mature cure. Any questions about any of that? All right. A characteristic of envelopes is that they can have enzymes on them. And that's not really surprising because the most important molecules of any virus are going to be on the outermost portion of the virus. And this is the portion of the virus that first sees the host cell. And that's why the virus, who's our new president, and not president, but Speaker of the House. anyways, he's the new Speaker of the House, and so he'll change things. And this is similar to a virus infecting a cell. It will use what it can the first time, but once it then uses up whatever the heck the virus was eating, I'm talking about it as if a virus is a person. So if the virus fits fairly well and the cord also fits, you can think about buying a shoe like that, or you could buy another shoe. One advantage of the specific virus shoe, which I showed you just a little while ago, is that the lab shoes, besides being made of leather, and leather protects your feet much better than many of the other agents we can treat a virus with. And I'll talk a little bit about that later. And then the last thing I want to talk about at this slide is that viruses can have a linear genome, or the virus can have a circular genome. And the sun could get in between. I might have a picture of that. Or the virus, meaning the sun, can do what it wants. And one of the things it'll do to grab control of the host cell is the virus will degrade the host cell's genome. Why does the virus want to degrade the host cell's genome? Anyone have a guess?
To infect it?
Say again?
To infect it?
Well, yes. My point was that viruses, when they infect a cell, they will inject the genome of the virus into the host cell and then use the host cell to make copies of the virus. And one of the first things the virus does is it turns on a gene for degrading the host DNA. Why do you think the virus might want to degrade its host cell's DNA? Any guesses? The reason why a virus will infect a host cell and take over the machinery of the cell is because a virus will turn on a gene which degrades the host chromosome. Why do you want to degrade the host chromosome? Because if you do that, then only your genome is in the cell, and then the virus, can dictate what the cell is going to do. If you have the host chromosome still working and still functioning, it would muck up the job, meaning your lawn, your grass wouldn't look correct. Any questions about any of that? All right, viruses can have their genome in a linear arrangement or a circular arrangement. For example, I think polio is an RNA virus, and its genome is in a linear arrangement. A better example would be the flu virus, and it is in a linear arrangement. But viruses don't have to be that way. Many viruses have their genome surrounded with the head, the protein capsid, and then that will be surrounded with a helmet, like a bicycle helmet. So prokaryotes have a linear genome. Eukaryotes, whoops, I didn't say that right. Eukaryotes have a linear genome. Prokaryotes have a circular genome. And viruses can be one or the other. All right, lastly, some viruses have their genome in several segments, like the influenza virus has its genome in several segments. That should not be confusing to you because humans have their genome in several segments. What we call these segments of DNA is chromosomes. Any question about any of that? Shrink that a little. We'll blow it up in a minute. Oops. What did I do? Oh, I made it way too small. Let's get that a little bigger. Oh, that's close. Let's go with that. All right, I mentioned that viruses always have a protein coat surrounding the viral genome. We call this protein coat the capsid. The capsid can be composed of subunits, capsomeres. Some viruses only have one capsomere, and I started to tell you that and got sidetracked. And you can see a capsomere right here, meaning this protein makes the viral coat the capsid, and it's only one protein, and this protein just repeats numerous times to surround the virus with the protein that we call a non-enveloped virus. And usually that's called a naked virus, meaning a virus that doesn't have an envelope is called a naked virus. And of course, if it has an envelope, we call it an enveloped virus. The envelope is seen only in some viruses, and it is a lipid bilayer. It does, or mostly a lipid bilayer. It does have proteins and carbohydrates in it as well. And that should not surprise you because the lipid bilayer that makes up the envelope of a virus is very similar to the lipid bilayer of our cell, meaning the cell membrane. The envelope is very similar to the cell membrane. The envelope is external to the virus. It's on the other side or the outside of the protein coat. If a virus has an envelope, it might have spikes. Oops, too far. This virus has both an envelope and a spike. The spike are those purple things. Spikes are very important for a virus if the virus has spikes, because usually the adhesion molecules of the virus are in the spikes. And then another reason why spikes are important is if your immune system reacts to this virus, the immune system will most likely be reacting to a molecule in the spike, probably a protein or a glycoprotein in the spike. Why that is the case is our host immune system always recognizes the outermost molecules of an invading microbe. And on a virus, the outermost molecules are on the spikes. Any question about that? So the spikes are important because this is what the host immune system sees with the virus, the spikes, if the virus has a spike or spikes. And then the second thing is the adhesion molecules are on the spikes, and that is how a spiked enveloped virus usually binds to its host cell from the ligands or adhesions on the virus, which is in the spikes. And when we look at that, oops, went too far. The adhesions I could have sworn I have that in here. I do not. All right, the adhesions are on the spikes, and so it's those molecules on the spike that'll bind to a receptor. I don't see where I have that now, but so what binds to a host cell if the virus never has spikes? Like this naked virus, what binds to the host cell? It's pretty easy. The adhesions are always on the outermost molecules of the virus. So it will be the capsid, the protein coat, that will be binding to the receptors on the host cell. All right, let's talk a little bit about, way too far again, about viral structure. When we're talking about general morphology of viruses, we're talking about the shape of the virus. Viruses generally have 3 different shapes, and we can classify viruses on their shapes. There are helical viruses where the capsid is in a helix. And then there are polyhedral viruses. This is a many-sided dice-like polyhedron. And so we can have helical viruses and polyhedral viruses. And the third one is a complex virus. A complex virus is shown here, here, and here, and that's the same one as here. You can tell that the complex virus can be essentially any shape. And this is just what virologists classify viruses by. Complex virus, meaning it's not helical, it's not polyhedral. All right, any question about that? So that is a naked virus that's complex, and this is a enveloped mammal virus. All right, you're looking at one of the most common viruses out there and the most studied virus of all viruses. And that would be bacteriophage T4. Remember, bacteriophage is simply a virus that infects bacteria. I guess I should state that viruses are generally classified to what kingdom they infect. Like there are animal viruses, they infect animals. There are plant viruses, they infect plants. There are viruses that infect protozoa, and there's viruses that infect bacteria. Any question about any of that? The reason why T4 is the most highly studied virus is probably just because T4 infects E. coli cells, a cell that was very common in the lab of early microbiologists. So early microbiologists started studying T4 because it was easy to grow. You just grew it in E. coli cells. T4 is of the T-even family. The T-even family has this type of a shape. Let me see if I've got that on the next slide. No. By this type of a shape, we're talking about this is a naked virus. It does have more than one capsomere, though, because there would be a capsomere for the head portion. There will be a capsomere for the sheath portion of the tail. The lower portion is the tail. And then there would be a capsomere for probably the tail base plate, as well as the tail fibers. And then, of course, the tail sheath. So this virus is made up of more than one capsomere, and that's unlike this virus, which is composed of only one capsomere. Any question about any of that? All right, so let's talk about viral infection and the host immune response. Infection with a virus, when it happens to an animal cell, the animal can stimulate antibodies, and these antibodies will work to attack the virus. So antibodies generally recognize and bind to viral surfaces, and that means, once again, the antibodies are binding to the surface of the virus. And what's the surface of here's my nice picture. What is the surface of a virus? Like a complex virus? The surface will be the outermost molecules. So on the bacteriophage T4, the outermost molecules will be molecules of the capsid, both the head and then the tail being the base plate, excuse me, down here, the base, and the mechanics. All right, any questions about anything? By morphology of the virus, we mean the shape. And we're looking at several different shapes here. That's bacteriophage T4. Oh, I already talked about that. Actually, I started talking about this one. So some viral proteins can change or mutate frequently. And if they do, then the antibodies will no longer work against that virus. An example of that would be the flu. Let me see if I can get this to work here. Maybe I'll open it from Word. Once you get a vaccine or get immune to a virus from getting infected, initially you'll have high antibody titers. Let me see if I can draw this. Here we go. I'm not able to draw on this. Let me see if I can get Paint open. Come on. I can, let me stop the share here and I'll get Paint to work. I'll begin the share in just a minute. Well, it started it. Let's get to open again. Let me delete that because I don't need it. And now I got to share my screen. So all viruses, when you first get infected or you first get vaccinated to the virus, initially you'll be producing, oh, that's not what I wanted. What am I doing? There we go. Initially, you'll be producing antibodies. And this is just the time on the x-axis and the number of antibodies on the y-axis. Initially, you'll be producing a large number of antibodies, but over time, this will begin to drop. when you drop below a certain threshold, they say you have no longer any protection from the antibodies. That isn't quite true, and that is the drop is gradual, and it's kind of arbitrary where you draw the line and say once the immune response drops below this line, that there's essentially no immune response to the antibodies. The point is that viruses can change, and an example of that is the flu. Once you become vaccinated with the flu shot, by about 3 years, your immunity is dropped to the level or less where we say you're essentially not protected anymore, meaning the flu vaccine isn't giving you any benefit, in which case you should definitely go and get another flu shot if you want to avoid getting the flu. But initially, your immune response is still strong, although it's decreasing. And this will go on for at least days, maybe weeks. And if it's endospores, it could go on for years. All right, any question about that? So the number of antibodies produced and the immune response being this line here, I guess. And then with more time, your immune response will weaken, which point at this point, you should get vaccinated again. Where that point is for the flu shot is at 3 years. If you had the flu shot over 3 years ago, then you should get another flu shot, because 3 years ago, the amount that the flu shot is going to protect you will be minimal. So you should get vaccinated again and then bring up protection. And we could state this, let's go ahead and state that usually protection is very good at the start, something like 95%. Oops, didn't get the 9 in there. But that will decrease as time. Another example of a virus that mutates and changes fairly rapidly is COVID-19. They haven't figured out exactly how often we should get a flu shot because, well, we just don't know yet. But it looks like you'll need to get a COVID-19 shot at the minimum every year. I do not know why COVID-19 appears to change more rapidly than the flu. It may be just because the threshold is higher because COVID-19 can kill people. And so it might have just a higher threshold, and that's the wrong one to do. But good enough. Any questions about any of this so far? That isn't true of all viruses. For example, the polio vaccine was made in the 1950s, and that's both polio vaccines. We're still using the polio vaccine today. So that vaccine, even though that was made, what is that, over 70 years ago, it's still good, meaning polio is not changing, at least not very rapidly. And so the vaccine is still functioning. Whereas the flu shot, we have to change it at the minimum every year. And actually the health professionals do change it every year. And certainly every 3 years. All right, so let's talk about viral taxonomy. Viruses are grouped into families based upon the type of nucleic acid they have, or on the replication strategy, or on their morphology. We can classify viruses like maybe one order higher than families. What the heck would that be? King Peter's son came over from Germany. So it would be order. So viruses can be classified into orders, but higher than that, we do not group viruses because we do not know what grouping the virus goes into. So we mainly have viruses by families, and then we have viruses into their genus, and then viruses into their viral species, and then we have subspecies. An example of a subspecies would be herpes simplex type 1 and herpes simplex type 2. Those are the same viral genus, herpes, and they're all in the herpes family. And it's the same viral species, herpes simplex, but it's a subspecies. Any question about that? Subspecies are extremely important in viruses. An example is one species of the poliovirus gives the disease polio. Another subspecies of the virus is the vaccine. Let me see, I believe it's the Sabin vaccine to polio can be used to protect against the strain or subspecies of the virus that gives the disease polio. Any question about any of that? All right, so subspecies are extremely important in viruses. The viral species is very important too. A species is a group of viruses sharing the same genetic information, and they live in the same ecological niche. Like herpes simplex type 1 and 2, their ecological niche is growing in humans. And that's actually true of polio, as well as the subspecies that we use for the polio vaccine. Genus names of viruses end in the name virus, like the herpes virus, that's actually a genus. And then the family name of a virus ends in viridae. For example, herpes viridae is the herpes family of viruses. Now, what you will like about viral taxonomy is we use common English names for grouping viruses. There is no Latin naming system. So herpes virus, like herpes simplex type 1, that is the scientific name as well as the common name. And poliovirus is the scientific name. And for those of us who are English speaking, that's the other benefit. They're always English names. And it's just that the early virologists were all English speaking. And then when other people started studying viruses that used another language, they went with the naming system that would already been started. Any questions about any of that? Hopefully that makes you happy. You don't need to learn any Latin names for viruses. You just use the common English name. An example of taxonomy, I already gave it kind of. Family, herpes viridae is the name of the family. The genus would be the herpes simplex virus. Common name, human herpes virus or HHV. And actually, this name has not been widely used by people. People are continuing to use the older name, the herpes simplex virus, which is the species. And then the subspecies would be herpes simplex type 1. Any question about any of that? All right. For growing bacteriophages, these are viruses that grow in bacteria cells. You just grow them up in a lab and you grow them in a bacteria cell. So bacteriophages can be plated on an agar plate that has had bacteria put down, and the bacteria are put down in the fashion so the bacteria will grow everywhere, and we call that a lawn of bacteria, and that's what makes the plate look white or off-white here. I guess that would be off-white. Okay, and then the bacteriophages will grow in the virus and create the plaque. So what happens is a bacteriophage touched a bacteria and then grew in that cell, and then it went out to infect other cells around it, and then it grew in those cells and went out from those cells, and that will give rise to a plaque. A plaque is where you see no bacteria on the plate. Let me blow that up just a little more. Here we actually have like 3 plaques together, but that plaque is much larger than, say, that plaque or this plaque. And probably what happened here is the bacteriophage immediately infected the bacteria because the two were so close they were touching. Whereas here, the virus was near the bacteria but not touching. You needed to have the bacteria grow before the bacteria touched the bacteriophage. And then once they touched, the bacteriophage could then start replicating in the bacteria. And that's why this plaque is probably smaller than that plaque. Any question about any of that? I actually grew bacteriophages like this in the past. All right, if there's no further questions, I think I'll stop here and we'll continue with this slide next time. All right, any questions? If not, I'll be in the lab from 6:30 to 6:45. Just answer questions. If you don't have questions, you don't need to come to the lab. Bye.